I just wanted to thank our sponsors who are CRE Reduced, CHU Nimes, Pfizer, MSD, Gilead and Baxter Healthcare as well. Thank you so much for your support, allowing us to proceed with this evening. So the final speaker tonight is Professor Jan Willem Alffenaar, who joins us from Sydney. So Professor Alffenaar is a chair of clinical pharmacy at University of Sydney, and his research interests include the use of anti-tuberculosis drugs, as well as the clinical application of TDM. And he's developing some implementation strategies for TDM in different settings to make the best use of the available analytical techniques, and also with a focus on patient-targeted approach. So thank you so much for joining us. I'll hand over to you, Professor Alffenaar.
Thank you for the kind introduction. Of course, in the last couple of talks, there were lots of questions and uncertainties, and hopefully I can shine some light on some of these issues. So the topic is, can we improve TB outcomes with TDM, and how can we design the studies to prove that TDM is beneficial for our patients? So, and when we think of improving TDM, we have to define what we consider improved outcome. And I think that is a very interesting question. If we look at the guidelines, TDM is already suggested for a lot of situations, and already elucidated on a couple of them. But if you look at them at this list, these are patients with complications. They are either not responding to treatment, have comorbidities, have reduced organ function, experience toxicity, drug-drug interactions. So it all makes sense to do TDM for these patients. But if we really look at the use of TDM, we know that it is underutilized. So what's happening? So this was a study, a systematic review that looked at TDM of the first-line TB drugs, and they systematically searched literature, and they found 41 studies reporting drug levels collected 2 hours after drug intake. Only 12 of those studies reported actual clinical outcomes, and based on that number of studies and the information found in the studies, they could not find a clear association between low levels taken 2 hours after intake and treatment outcome. They mentioned that the limitations of these studies were that actually most studies did not report therapeutic outcomes, so they only looked at levels and target attainment. And actually, if levels at 2 hours after intake may be low, they may be higher at another time point during the day if a patient, for example, is a slow absorber of the drug. And there are also different types of studies, and this is an example of what we did in our TB reference center in the Netherlands. And because we did this study, I can be extra critical. In this study, we collected data on about 80 patients with MDR-TB and looked at the aminoglycosides. And of course, we know this is a very toxic drug. So a long time ago, we already started doing TDM for this drug, and we aimed at a target of Cmax over MIC of about 20, which we derived from other infections like cystic fibrosis, where you need a higher exposure to get to the target site and kill the more resistant pathogens. So we wanted to be safe, and we aimed at a little bit higher targets than you would normally do with aminoglycosides. And when we looked at the results, we found an average Cmax over MIC of about 25. We did do a lot of monthly audiometry, so we could actually look at hearing loss, and we found that we had only 11% hearing loss versus 40%, as was published over 2 decades ago by Pelican and CID at the standard dose. We also looked at relapses, and we were not able to find any relapses in our patients. But there are, of course, lots of limitations in this study. Well, for example, it was a single center study. So apparently we selected patients for TDM and we did well. However, it's also retrospective. It was over a period of 12 years. So likely in that study period, some things may have changed. We had, of course, missing data. We had loss of follow-up. TB patients in the Netherlands are mostly asylum seekers, so they travel or they have to go to another European country. And of course, we lacked a control group in our setting. So although it seems very favorable for TDM, we actually cannot make strong recommendations based on this information. Also, if you look at a global level, TDM is mostly done in patients with a poor response to treatment, despite being adherent to treatment, having received DOT therapy, or because of toxicity. And of course, we know that some of these older TB drugs are very toxic. But of course, many patients are also managed in another way. If we look at first-line drug, treatment can be extended if patient is not responding. Sometimes people add an injectable streptomycin. Drug susceptibility testing is repeated. If we look at drug-resistant TB, of course, poor response is more, let's say, accepted than with drug-susceptible TB. And as Andrew already talked about, dose reduction and interruption is very common in linezolid to manage toxicity. And last but not least, of course, aminoglycoside stopping the drug, but of course that reduces the treatment of your intensive phase of MDR-TB treatment. And when actually TDM is performed, it's often only a C2 level. So the implementation hurdles for TDM there are plenty. Not only we lack lab facilities, there are, for example, no immunoassays for most of the anti-TB drugs. And if you require advanced assays, this is also a challenge. We know about sample stability of the first-line TB drugs and, of course, training of our staff, funding. But I think evidence and cost effectiveness are perhaps the most important ones. So if we want to improve TDM, we have to look at efficacy and toxicity, and we have to look at what we achieve with standard care. And if we look at the traditional therapeutic range of the drugs, this completely ignores pathogen susceptibility. It's only about the levels. So more recently, people start thinking about AUC over MIC, and that, of course, takes into account pathogen susceptibility. But the biggest problem is that the MICs are not readily available when you want it. It takes time. And if we look at most of the studies, these are actually not TDM studies. They're mostly a kind of observational drug exposure study looking at what happened with the patients, but no interventions. And I think that is very important if we talk about TDM, the actual intervention based on drug concentrations, clinical condition, and microbiological results. So if we are thinking of what type of outcomes could we include in our TDM studies, we have, of course, the short-term outcomes, let's say 2 weeks and 2 months sputum culture conversion. We could think of end of treatment for drug-susceptible TB, a 6 months sputum culture conversion in drug-resistant TB. And if we really think of long-term outcomes, end of treatment for drug-resistant TB or relapse-acquired resistance after treatment. But that already takes a lot of time to collect your data. For toxicity, it may be a little bit easier, especially if we talk about toxic drug like linezolid. But again, we need to have regular intervals during treatment to monitor our patients. So if we want to use TDM to improve the efficacy, we have to at least consider the PK/PD targets which we know, because for some of the drugs, there's no proper information or adequate information to actually drive our dose decisions based on drug concentrations. And of course, we should not forget about the costs. And if we know that we give 5 drugs for MDR-TB or 6 drugs, what happens between synergy and antagonism in that regimen, and what does that do to your target concentrations? So if we consider all these variables and we have to decide a primary endpoint for our study, we have to be very mindful how to select that endpoint. Because if we want to go for a short-term endpoint, let's say 2 weeks, we have to increase our frequency of sputum sampling to actually show a difference between patients. And if we look at long-term outcome, for example, the traditional MDR-TB treatment of about 2 years, a lot can happen during treatment. And how can we be sure that what happens can be attributed to MDR-TB? Especially if we want to show that TDM works, we should be having a timely intervention. Because if we send up samples and we get results a month later, I don't think that will really impact on practice. And again, I would like to emphasize that we need that pathogen susceptibility to actually select the most appropriate dose for the drug. Already elucidated a little bit is what we do about the levels. And this is an example of rifampicin. And on the left panel, we can see the distribution of the peak concentrations in a study. And we can see that the peak was not at C2 for most patients. Only 20% of the patients had a C2 level that was actually Cmax. And so peak levels were either earlier or even much later, 6 hours after intake of the drug. In a more detailed figure on the right, you can see how these curves can differ between patients. And if we look at the C2 level, there's a patient that didn't start absorbing the drug, and there was a patient already having the peak at 1 hour after drug intake. So a C2 level may be practical for a routine care perspective, but for a clinical trial, trying to explain what's happening to a patient and what drives outcome, I think it's not suitable enough. So we need to find better ways to assess drug exposure. And of course, we can collect a full PK curves, which means around 8 samples. And if we talk about drug susceptible TB, it's at least 4 drugs. And I estimated $30 a sample, but Andrew mentioned $100 a sample. So you can see that it is already consuming a substantial amount of money from your study budget if you want to do a large-scale TDM study. And then we have to talk about the cost for the research nurse. So I think such type of study is only feasible, at least when we have multi-analyte assays that can help to significantly reduce the assay costs for such a study. Of course, limited sampling strategies may be helpful, but if we want to optimize 4 to 6 different drugs, we're really lucky if we find a limited sampling strategy that can be found suitable for all these drugs. And then again, the sampling strategy has to be appropriate for the specific population. We know that with a genetic variability or other variabilities, limited sampling strategies may not fit each population. So I think that with Bayesian dosing, we can do a little bit better than with linear regression. So if we use our population data and are able to individualize based on individual patient characteristics and our TDM samples, I think that is the step forward in personalizing the dose in TB studies and TB treatment in general as well. So I'd like to discuss some of the strategies based on this study. It's a study we did together with our collaborators in China and from Karolinska Institute in Sweden. And in this study, we were able to perform adequate PK sampling with full PK curves. We did actual MIC measurements for all the drugs. And we looked at treatment outcome at 2-week, 2-month sputum culture conversion and end of treatment outcome. We looked at acute kidney toxicity and liver injury. And this was a prospective observational study. And we defined two cohorts, a development cohort and a validation cohort, to verify the targets. So of course, you get large amounts of data on AUC over MIC. Of course, we have this combined with the measured outcome in all these patients with respect to sputum culture conversion and treatment response. And we analyzed this with a classification and regression tree analysis strategy. And that means that this is a kind of let's say, data-driven analysis. So the data shows you what type of target concentration may predict the outcome best. So it looks like this. In this case, we look at treatment outcome at the end of treatment. And these were the numbers: 16 patients failed treatment and 152 patients had successful treatment. And what drove this difference was having a rifampicin AUC over MIC over 435. And if you look at the patients with a value over that range, there was about 97% success rate. And if you looked at treatment failure, there was substantial more failure in patients having a level lower than the target. But of course, these targets depend on the actual population. So they're not easily transferred to other populations. This is another example from this study where we analyzed time to sputum culture conversion. So pyrazinamide appeared to be very important in this study, with a best predictor for 2-week culture conversion and 2-month culture conversion. And if the patient had a value between those two, it nicely demonstrated that it could predict a time to sputum conversion based on these 3 different values. So it's a complex strategy to analyze such a data set because, of course, we have a lot of AUC and MIC data, and we define targets, and subsequently we have to include other variables as well as we know TB treatment outcome is not only determined by drug levels, but also by adherence and by smoking habits, etc., etc. So we all have to take into account those variables as well in the development of the analysis. And what we subsequently did was use the development targets and see whether these targets were reproducible in the validation cohort. So this was an independent cohort of patients treated for the same disease. And when we compared the ROC curves for all the individual outcomes, they were all in the same ballpark, showing that in this particular Chinese population, the targets as derived were reproducible in a comparable population. So when it becomes interesting is when we want to define a target for a drug. So in this case, for example, for rifampicin, we had defined AUC over MIC over 435 for efficacy. However, for toxicity, the value had to be lower than 82. So that means if you start comparing what is feasible and what is toxic, and then you end up with an MIC that is treatable with that particular exposure without causing too much toxicity. So there is a balance in what we can achieve. And of course, our recommendation was that these results from this observational study needed to be confirmed in a randomized controlled trial. So this is the highest level of evidence to support TDM. We have our randomization, we can control, we have our standard of care treatment from WHO. And of course, we can do lots of observations in a clinical trial. We can define our participants. And of course, we would like to do some blinding. However, if we want to blind a TDM study, it becomes a challenge because we have to do sham procedures for TDM blood sampling. We have to use lots of placebos in case we want to make dose adjustments. So that is a bit of a hassle, and especially if we talk about the number of drugs, 4 drugs in first-line treatments and 5 drugs in resistant treatment. So it is not easy. And of course, we are waiting for MIC results if we want to adjust the dose. So a lot of challenges. So at this moment, I think this is mission impossible. So when we move on to, let's say, open label studies, although these are considered producing less rigorous evidence, I think we can correct for a lot of the potential biases in such a study by using, of course, appropriate measurements, by using direct observed therapy, and using rigorous treatment protocols. So if we move on, we could say we could target AUCs of key drugs. We could use fast genotypic DST to select the most appropriate drug, and then of course look at treatment outcome in specific populations. And it becomes interesting when we are going to select that population, because as already mentioned, the success rate of drug susceptible TB is quite high. So that would require a large sample size. But if we move on to more resistant populations, it's, let's say, likely that we can use a smaller sample size. It becomes very interesting when we could actually target patients at a low exposure. So if we can use that information in a kind of let's say, simulation scenario, we could consider that we need to screen for this low exposure and either subsequently have this as a intervention arm where we screen and adjust the dose and have control standard of care. Or what we could actually do is pre-select patients with low exposure and then randomize them for dose adjustment or standard of care. That would significantly reduce your sample size, but we don't know yet whether it is ethically acceptable by the committees. If we're looking at MDR-TB, of course, assays are required, being a bit more sophisticated because we are using so many drugs. And of course, we could work with drug susceptibility testing with target concentrations, let's say 1 dilution step below the breakpoint, and adjust when results become available. Of course, outcomes should be targeted either at 2 or 6 months sputum culture conversion, and it depends a little bit on the site. So in some countries, average sputum conversion is at 6 months, while in other countries it's much sooner. So to summarize, could TDM improve tuberculosis treatment? I think we can say yes. We have lots of observational data showing all at the same direction if we look at that data, but we need that real world study where we combine drug exposure, pathogen susceptibility, and outcome. We should aim for a feasible trial design because we want to implement in practice later on. And this means that we have short turnaround times. And of course, we can use slightly different designs for different situations, looking at drug susceptible TB or MDR-TB. So I think for now, we should keep on dreaming of our double-blind TDM study. And with that, I would like to end the presentation and happy to take any questions.
That was wonderful. Thank you, Jan Willem.